I am now pleased to introduce our next session, Effects of Transferring Gut Microbiota Through Co-housing on Bone Health and Immunity of Broiler Chickens, presented by Bilal, McGill University. Take it away, please.
Hello, everyone. My name is Bilal, and today my talk is about the importance of gut microbiota, and especially in reference when the gut microbiota is transferred from the older age birds toward the young chickens, and to show and to study the effects of such treatment on the health of the young chickens in terms of immunity and bone health. So I will pass through a little bit about the background, which is related with the importance of the gut microbiota, the rationale behind my study, and how I performed that study and the results. And at the end, I will describe the summary of my study. So here I'm going to show a couple of slides, which actually shows that chicks can be reared in two different environments. One with the elder chicken, and the other one is our commercial poultry, where the chickens are reared all alone. So here we can see that when the chicken are in close contact with the hatching parents, so they are exposed to so many different environmental factors like the animals, flora, grains, and dirt, and thus it is exposed to different types of environmental microbiota. Although the environment looks dirty, but the exposure to the microbiota is a lot. On the other hand, in our commercial poultry, we use a lot of disinfectants and antibiotics, and we concentrate on the cleanliness of the environment to avoid any type of infection. So in this scenario, the environment is very clean, but the exposure of the microbiota toward the chickens is relatively less. So recently, a study described that while comparing the young chicks when they were reared with the hen and without the hen, and they found that there are 45 genera were in high abundance in the group of chicks which were reared with the hen, compared to the chicks which were not reared with the hen, and only 8 genera of microbiota were seen abundant in this group. And interestingly, 3 out of 8 genera showed that they have the pathogenic ability to have a disease. So the results shows that actually the beneficial or good microbiota actually, if present, it can suppress the other pathogenic microbiota, which is not available or not present in our commercial setups. So in our commercial setup, the microbial diversity is relatively sparse, which lead to the underdevelopment of microbiome. And we know that the microbiota are directly linked with the development of different host systems that include the immune and gut and skeleton systems. So this actually increased the vulnerability of the chicks toward more infections, and there are chances of pathogen localization. And to avoid that situation, we use more clean environment with the help of disinfections and use of antibiotics, which further reduce the microbial communities in the farm. And thus this lead to a vicious cycle where we try to achieve a clean environment on the expense of reduced microbial diversity. In our recent study, we have observed that we can improve the diversity and maturity of the chicks' gut microbiome with the help of probiotics. So We also seen that not only the diversity, but the maturity of microbiota can be increased, and maturity in terms that chicks acquired a microbial configuration early in age, which is actually seen in the older birds. And we also observed that these microbiome targeted therapies actually improved the gut health as well as the immune response in those chickens. So that lead us to have a hypothesis that if the microbial maturity improve, the health benefits, confer health benefits to the chicks. So if we transfer the mature microbiota from the older birds, we are hypothesizing that it will affect the health of the young chickens. So we design an experiment to see our hypothesis. We are actually in first step, we reared young chicks to 2 different age groups at day 14 and day 42. And after rearing, we acquired another group of birds, and we divided that into three groups. And after that, we mixed or donate birds from the older groups of day 14 and co-house them with the young chickens. And similarly, from the other group, we also co-housed the older hen with the young chickens. And later on, these birds were reared for 42 days, and samples were collected from these young chickens at day 14 and day 42 to analyze different parameters like the T cell immune response, the analysis of fecal microbiota, and the tibial bone health. So as an overall, we couldn't find any significant differences in terms of production, and the body weight, feed intake, and FCR were similar across all 3 groups. But when we observed the fecal microbiota, so we observed that at early age, at day 14, there were not very significant differences. But later on, at day 42, we observed that there are significant differences across the groups, and the microbiota started to cluster differently. So we also checked the alpha diversity, and we observed that in the B42 group, the alpha diversity was significantly high in the B42 group. And similarly, when we observed the beta diversity, so we have seen that by composition, the microbiota in A group as well as B42 group was significantly different from the control group. So that shows that the treatment effects actually affect the fecal microbiota, and it's improved its diversity. Interestingly, when we further checked the alpha diversity, we found that the significant effects were not in terms of richness. The observed OTUs were almost similar across the 3 groups, but interestingly, the evenness was significantly high in the B42 groups, which show that microbiota in this group was more resilient and more even and more stable. So regarding the relative frequency of different microbiota, we observed that the firmicutes were relatively high in percentage in B42 group, and this is the phyla which are considered good extractor of energy from the feed, and it includes different short chain fatty acid producing families like Lachnopharmaceae and Ruminococcaceae. So further, we checked the T cell immune response, and we observed that the CD4 cells in the B42 group were significantly low as compared to the other 2 groups. But we observed more interesting things, that the T regulatory cells were significantly high, and we know that T regulatory cells are actually responsible for the maintenance of the anti-inflammatory state in the gut. So further, We observed the CD8 positive cells, but we couldn't find any significant results. So the effect on the T cell response, it shows that the treatment, especially in day 42 group, B42 group actually skewed the immune homeostasis toward an anti-inflammatory state. We also observed significant differences in the bones, and we seen that the tibial bone length was significantly increased as compared to the control group. And we also observed that these significant differences were also in terms of bone mineral content, which were high. And similarly, the bone pore volume was significantly less in the treatment groups. But these observations were early in B42 group, which were the older donor groups, as compared to the A group. We also conduct the latency to light test, which is usually checked to see the lameness. Here we couldn't see significant differences, but although the trends were there in both two groups, some improvement has been seen in both two groups. So in summary, cohousing day-old chicks with 42-day-old chicken improved the alpha diversity of fecal microbiota and skewed gut immune homeostasis toward an inflammatory state. And cohousing improved the tibial bone length and the cortical bone mineralization and porosity, and which we consider that this will help in the improvement of welfare of the birds in terms of bone health. And further, these beneficial effects were more prominent in the older donor groups as compared to the young donor groups. So thank you so much for your attention, and I would be happy to have your questions.
Thank you very much, Muhammad. We have about a minute and a half for questions. Reminder, you can put your questions in the Q&A box. Very quickly, I think you mentioned that you measured mortality. Did you see any differences among your treatments with respect to mortality?
Yeah, initially we were worried that, and we were expecting that there would be mortality, but we couldn't see mortality because of any infection or something like that. But in one group, the day 42 group, which were the bigger size of birds were donated, there we see in first 2 days a couple of birds were squished because the experiment was in cages. So mortality, we couldn't find any mortality because of the treatment, but just in a couple of occasions because of the birds were young and space was smaller, so they were squished there, but no mortality because of the treatment.
That's very interesting. I have more questions, but I think we're going to have to leave it there, as we will need to move on to our next session. Thank you very much.
Thank you so much. Thank you.